Madhu, Madhu, close the door.
Yes, sir. I think we can begin. People are. It will cross. It will cross 60. It will reach 100 by the time it becomes 240. So I think we can start. I don't want John Lough sir to keep waiting. It's already early morning.
No, it's okay.
Yeah.
I warn you, I'm not a morning person.
Okay. Good afternoon, one and all. It is said that
Yes, good afternoon.
when God gives us a new beginning, it starts with an ending. Be thankful for closed doors. It starts because they often guide us to the right one. Today, I'm thankful to God for having given us the gift of life and strength to deal with this COVID pandemic. Many of our doors are closed, yet opportunities creep in. One of the biggest gifts of 2020 is this particular session with Dr. John Lough. It has been 17 odd years that I met him or mailed hardly 3 or 4 times during this gap. Yet, he agreed happily to grace this occasion with us and give this lecture. If I were to virtually column each of my acquaintances, then Dr. John Lough would elute out into fraction of the kindest and very good human I have ever known from England. I welcome you to this session, sir.
Thank you.
Dr. N.D. Satyanarayan, Associate Professor, PG Center, Kadur, Kuvempu University, Karnataka, will be chairing and moderating this session. Dr. John Lough happens to be his postdoctoral guide at University of Sunderland, UK. He is having MSc degree in pharmaceutical chemistry and PhD in isolation and synthesis of anti-ulcer drugs, both from Gulbarga University. He did his postdoctoral studies from University of Sunderland, UK, and was a subcontract researcher at GlaxoSmithKline. He joined Kuvempu University as assistant professor and is at present associate professor, director, chairman, and coordinator of PG Center, Kadur. He has one national and another international patent to his credit. 50 plus publications in peer-reviewed journals, both national and international, and he has successfully guided 8 PhD students and 7 are presently working for it. His areas of interest and expertise are separation and identification of natural products and synthesis of biologically potent small molecules for anti-ulcer studies. He was awarded with British Council Fellowship and then Postdoctoral Research Fellowship from Stifel Laboratories, Bursary Fellowship to visit Italy, and for York Center. He has attended courses on bioanalytical assay and natural product separation and LC troubleshooting and LC method development and validation. He is also a life member of Chromatographic Society of India and Indian Academy of Science. With this brief introduction, I welcome Dr. N.D. Satyanarayan to chair the session. Welcome, sir.
Thank you very much, Pratima.
Our principal, Dr. S.E. Nataraja, is here with us. I now request our principal to formally invite both our speaker as well as the chairperson to this session.
Thank you, Dr. Pratima, and good day and good afternoon, to Dr. Lough and Professor N.D. Satyanarayana. It's a great pleasure to be a part of this webinar. I'm extremely thanking that two gentlemen, especially Dr. Lough from UK, Sunderland University, and has taken much time, take out time, and has agreed, and today is delivering a talk on chromatographic and allied subjects. I'm extremely thankful and extremely grateful to listen to him. I thank him wholeheartedly from myself, from the Department of Chemistry, Department of Collegiate Education, and of course, from the government of Karnataka, for giving, exposure regarding, chromatography for the students and for the, whole, participant that are there in this webinar series. Thank you one and all that. I hope and I wish best outcome will come out of this session. Thank you one and all. Thank you.
Thank you.
Thank you, Nataraj sir.
Thank you.
We have our head of the department also, Dr. M.P. Somshekarappa sir, here with us. I welcome him. And now I hand over the session to Dr. N.D. Satyanarayan.
Thank you very much, Dr. Pratima Matthias, for your introduction and also inviting to chair the session for today's International E-Conference series on advances in chemical sciences, health and education. Ladies and gentlemen, I have the honor and privilege today to welcome and introduce one of the world-renowned speakers for today's lecture, Dr. W.J. Lough from School of Pharmacy and Pharmaceutical Sciences, University of Sunderland, UK, who is a fellow of Royal Society of Chemistry and Chartered Chemist of Royal Society of Chemistry and a fellow of Chromatographic Society of UK. To begin with, Dr. Lough obtained his BSc from University of Glasgow, UK, a PhD from the Department of Chemistry at the University of Strathclyde, and completed 2 postdoctoral fellow research from University of Edinburgh and City University London, where he worked on iron pair HPLC. Later, Dr. Lough joined as senior scientist at Beecham Pharmaceuticals as leader of chromatographic team supporting drug discovery and preclinical drug development, working on drugs such as nabumentone, chromocalin, and paroxetine, of which he was the first person to carry out chiral separation of paroxetine, wherein he and his team was instrumental in bringing the drug to the market. Dr. Lough has wide experience of industry and research, which he is currently bringing to bear in order to make a contribution to enhancing teaching on undergraduate and postgraduate pharmaceutical sciences courses at University of Sunderland. At his university, he is the program leader for MSc Drug Discovery and Development and teaches widely across the BSc for Biopharmaceutical Sciences course. Both of these are excellent models of how employability ought to be embedded into an academic course. Over and above his extensive teaching and associated administrative duties, he is actively involved in research, wherein he has published more than 80 research publications of high repute in one of the most important journals in the globe. He has delivered more than 100 invited presentations and successfully guided 33 students for their PhD degrees. And he was also instrumental in editing and co-editing 5 books, wherein these books are used worldwide, which include chiral liquid chromatography, pharmaceutical and biomedical applications of liquid chromatography, HPLC, fundamental principle and practices, chirality in the natural and applied sciences, and drug geochemistry. Dr. Lough has obtained £780,000 in external research funding and made contribution to successful departmental funding bids in excess of £2 million. He is a long-standing member of the Chromatographic Society. He was the president of Chromatographic Society of United Kingdom from 2007 to 2009, and he is an active member of British Pharmacopoeia Expert Advisory Group, MC1. He has delivered short courses nationally as well as internationally in countries like USA, Canada, Brazil, and not to mention India as well. He's consulted widely in the United Kingdom on chiral patent cases in North America. He chaired 8th International Symposium on chiral discrimination and served on 7 other international symposium committees. He's, he has served on as editorial advisory boards on 6 international journals and currently referees for 12 international journals. With this brief introduction, I welcome Dr. W. John Lough to deliver his lecture and enlighten us all.
Thank you very much, Satya. As always, I'll do my best to enlighten you. So one thing it didn't say is I'm not a master of IT, so hopefully this works. So I'm going to open a window so that hopefully I could still see you in the background. Yeah, okay. So good. Right, hopefully I could hear you, so just shout if you stop seeing the slides or if my internet causes a problem. I think we should be okay for the time it takes to deliver this lecture. Okay, so this is the title slide, The Impact of Chromatography in Industry. And clearly I've picked the pharmaceutical industry because most of my chromatography I've done has been involved in the pharmaceutical industry and in drug analysis. Now, I've chosen this rather than working on some really advanced piece of research in chromatography, because my experience of delivering lectures to a wide audience who aren't chromatography specialists is that the main thing I have to do is to convince them that analysis, and in particular chromatography, is really important, and it's not just something that the QC guys do and you send in a sample and you come back with a number. It's much more than that, and it has a major influence in how industry operates and how you get products to the market. So that's why I've chosen this topic. So hopefully you'll agree with me that it is suitable for a wide audience such as yourselves. Okay, I'll start by saying where I am. I like doing little maps, and this is the UK, and I use this one just about every time I speak outside of the UK, just to reassure people that Sunderland isn't an outpost way up in the northeast of England, but it's very much bang in the middle. And we like to think we're the centre of all the activities in the UK in the teaching and research of pharmaceutical analysis. I have visited India quite a lot, and one of the things I liked doing was actually drew a map of India with the Western Ghats showing Kuvempu University and Gulbarga University. And I know exactly where you are because I believe I have visited it on a trip to Belur one time. But it's just a bit context. I haven't worked out how many miles away I am, but I think we're 5.5 hours time-wise. So I just had my breakfast. Okay, so that's where I am. And before I go any further, I'll stop with a quick advert. As well as doing lots of research still, I have to do a lot of teaching, and part of that is on a course called MSc Drug Discovery and Development. I just thought I'd mention that because I believe there'll be a lot of people in the audience who are studying towards a BSc. And if you're doing that, you could do worse than thinking about coming over to the UK to do research, to do a PhD, or to join up with this MSc course, which is one I lead. I mean, I teach pharmaceutical analysis, but I spent a fair time in industry and working with people in industry. So this course not just gives you drug design and toxicology, clinical trials, and pharmaceutical analysis, pharmaceutics, we're also steeped in the regulatory background. So if you want a career in the pharmaceutical industry, give this a thought. It'll be nice. It's always nice for people to go and visit another country. So I like to think that all the Indian students we've had over here in the years have benefited quite a lot by doing this course and returning to India. And a lot of them have got fairly responsible jobs now in the Indian pharmaceutical industry. A lot of them go into regulatory affairs and pharmacovigilance. Okay, so that's the adverts over. Right, so what I'm going to talk about, I'm going to talk what's it all about. I want to say a little bit about chromatography, not assume that everyone knows exactly what chromatography is, but just to emphasize one or two fundamental points. And then the main part of the talk is it's actually one or two selected examples. I've not crammed in everything. I've just tried to pick a good illustration for each example, the different stages in industry of getting a product to the market. I've picked the different stages, and hopefully you'll agree that the examples I've picked highlight the very influential role in analysis and getting things to the market. And the last part of the talk will be more about chromatography itself and how, by getting speed in chromatography, we could do a lot to enhance it. The speed in chromatography indeed is critical in getting speed to the market. So if you've got a drug and many other types of products, you're going to be needing to do a lot of chromatography before you get that product onto the market. And I'll say a little bit about current and future directions, what we're doing with research just now. And maybe sometime in the future, you might invite me back and I'll talk more about the sort of latest research we're doing. Okay, but we need to try and convince everyone that chromatography in industry is really important and plays a major role. So that's what we're trying to do today. To take you back to chromatography, when students come off our courses, they might want to go and be a synthetic chemist or do medicinal chemistry or even pharmaceutics. But what they find is that they want to get in industry. Most jobs are in analysis and also doing chromatography. And here's why. There are other techniques which are very expensive and influential, like NMR and mass spec. But in terms of widely used techniques that are readily available at a sort of affordable cost, chromatography is very much the most widely used technique. And you may be familiar at undergraduate level with simple techniques like UV spectrophotometry and acid titrations. So I just got this little slide here which always show our students to emphasize how chromatography is a step better than that, and why in most instances you'd use chromatography instead of these traditional techniques. Now, when you're developing an analytical method, it has to be you have to do analytical method validation, which means that the method is suitable for its intended purpose. And to show that it's suitable for its intended purpose, you have to do lots of tests on the method. You have to have a linear response for the detector response to the concentration of the analyte in solution. You need to have robustness, that the method will deliver the same results each time if it's subject to minor changes in experimental variables. You need an appropriate limit of detection and limit of quantitation. If you're looking at trace amounts of your analyte, i.e. the thing you're trying to analyze, if you're doing automatic sequence and your drug or analyte is in solution for a long time, you need stability in solution, and you need precision to show that you're going to get the results you get from repeat experiments are always going to be very close to each other, and you need accuracy that the result is close to the true value. And I've kind of highlighted in red there accuracy and specificity for the very good reason that you can get accurate results if your method is not specific. So the definition of specific is that a specific method means when what you're measuring comes from the analyte and only the analyte. Remember, I'll use that word a lot. The analyte is the species you're trying to measure. So I'll just show you an example of somehow the traditional analytical methods that you might use widely yourselves are kind of deficient when it comes to specificity. If we take some really simple examples, this structure here on the top left is it's an ester, and it's in fact that would be a good prodrug for a non-steroidal anti-inflammatory, and to one of its degradants you'd expect to be this aryl acetic acid. Not aryl acetic acid you'd expect, it's very similar to naproxen, a well-known anti-inflammatory drug, so you'd expect this to be an impurity in this. So if you're using UV spectrophotometry to quantify how much of this was in the sample, you'd be measuring what is absorbed by UV light. The chromophore is this naphthalene ring, and as you could see clearly, the naphthalene ring is still present in the degradant. So if you're measuring a solution using UV spectrophotometry, you'd be measuring all species that contain that naphthalene ring with the oxochrome on here enhancing the UV response. So it's not specific. What you're measuring is all of these, the compound and its structurally related impurities, not just the compound itself. And similarly, it's kind of even more obvious with acid-base titrations. If you've got this basic compound here, clearly not a drug, but the same would apply as a basic drug. I've put in a compound where I know the simple chemistry, what the degradants would be. You'd lose the elements of water to get a double bond here by dehydration, or you could get a secondary alcohol, you could oxidize up to ketone. So these would be potential impurities in this compound, and if you did a titration, you wouldn't know that these were present because they would react with your acid in the same way that this would. So neither, not just in this instance, in many instances, that these are examples I've picked to show it very obviously, but if you're looking at structurally related impurities, they've got similar acid-base properties and similar UV absorbance properties to the main thing you're trying to measure. So specificity is a problem. So how do we get around that? Obviously, we go to chromatography. And so chromatography is a differential migration process. You've got a stationary phase and a mobile phase. It could be a flat bed, like in thin layer chromatography. And there are lots of different formats. It's not just column chromatography. But the idea is that your liquid goes over the solid and you inject or apply your sample solution, and the components of the mixture, they go down the column carried in the mobile phase. They equilibrate between the stationary phase and the mobile phase, and if they've got a high affinity for the stationary phase, they take a long time to come off the column, if it's a column we're dealing with and not a thin plate. So they get separated because they come off at different times, because the position of the equilibrium between the analyte in the mobile phase and the stationary phase is different from that for the structurally related impurities. So you get a separation, and I've been in India and I've been in lots of labs where you've got these big glass separation columns, still used by synthetic organic chemists and also by people who are extracting active species from medicinal plant extracts. Okay, so this isn't a modern HPLC system. I use this one in teaching because it's the kind of systems that we're using, well, a modular system. I'm using that because you can see the different parts of an HPLC system. You have a mobile phase, which is situated above a pump, because you need high pressure to pump the liquid mobile phase through the column. The column contains very small, typically 5 micron particles, so it's not like a big glass column you have in a plant extract laboratory, so you need high pressure to pump it through. The pump delivers a continuous flow, pulse-free, down the column, and because the column's under pressure, you have an injector to inject your sample. Typically it'd be 20 to 10 microliters of the sample would be injected in the column. Column could be anything between 5 centimeters to 25 centimeters and might typically be 4.6 millimeters ID wide. When it comes off, you actually go through a detector. Most drugs absorb UV, so it's typical to have a UV detector, and in there is not a 2 mil cuvette, but a 10 microliter flow cell. But what it shares with the traditional UV spectrophotometry is that a 1 centimeter path length. So as compounds come off, they absorb UV light, and that gives you a signal where it's really a change of UV absorbance with time, and also the liquid goes to waste, and we worry about the environment, so we dispose of that safely. Okay, and all the signals coming from that detector go to the PC, or nowadays that could be a big laboratory information management system, and it would often control the pumps and automatic injectors as well. Okay, so that's what it looks like in a simple schematic terms. So what's the output look like? There's a simple one of say if we were wanting to isolate, measure peak 2. Clearly, if we wanted to be specific, if you see here, if it's not 100% baseline resolved, then we're getting a contribution from this. So to have a specific chromatography and measure this, it needs to be resolved from this. So you're trying to get resolution of peaks, like a good separation, in as fast a time as you can manage. And to do that, I've cut out some of my chromatography fundamental slides, but simply if we increase capacity factor, if we increase the retention time by changing the mobile phase, and it comes off in a longer time, we'll get separation. But we don't want to do that because the quicker you could get the separation, the better in terms of productivity. We could do some mobile phase optimization or change the column and get the ratio of these two retentions to be more different, and that would help. But also a lot of the progress in chromatography is about improving the efficiency. So the difference between the classical glass columns that you use preparatively and these analytical HPLC columns is the particle size. And if you use a small particle size, hence the high pressure is needed, these peaks, if it's efficient, smaller the particles you go, there'll be less band broadening in the column. These will get narrower and you'll get better resolution. So a lot of the developments in chromatography are about getting better efficiency and improved separations. And the narrower the peaks, the more peaks you could separate in a short unit of time. So that's essentially what we're trying to do with chromatography. The instruments are still fairly cheap. An NMR might cost $500,000 to $1 million, a mass spec maybe a bit less than that. But the HPLC systems we use cost £50,000, which is very expensive. We've got top of the range HPLC systems. I will not, unable to convert it to rupees and lakhs for you. I do apologize for that. But you can get a cheap system, a refurbished system, for about £10,000. And we were able to share some HPLC systems with some of the universities in India. And this is the kind of output you get. This is something that's quite common in the pharmaceutical industry. You've got a drug, you can measure the main peak if you want to get the purity, but these are structurally related impurities of this particular drug, acetaminophen, which is another non-steroidal anti-inflammatory drug, and you're trying to get this kind of separation. And you can see this one runs very close. To separate this impurity B from the main peak is really quite challenging. So chromatography is not just important, it's actually, for this difficult type of separation, it's quite a challenge to do it efficiently and effectively as well. Okay, so that's quite simply what chromatography is and why it's got a major role, because of this specificity. And a lot of the techniques you might be familiar with just simply aren't sufficiently specific. Okay, so now I want to get on to some illustrative examples of how chromatography has played an important role. And this in some ways is almost like a trip through history or a trip through my career. And I was trying to think, well, what are the best illustrations of this? Now, in my time at Sunderland, it's an academic institution, we've done lots of things which would contribute to industry. But I was thinking, well, what's the best examples of this? It's actually when I did work in industry, or when I worked for industry, and this takes you way back in time, but these are real examples where chromatography has had a major impact on a particular company or industry. And then towards the end, I'll talk more about chromatography itself and what we're doing in at Sunderland to help enhance speed in industry. Okay, so I'm going to start going way back in time. This is the only example that's not a drug industry. Oh, sorry, my apologies. This is pharmaceutical industry, drug discovery, preclinical, when you do your formulation development, your chemical development, your toxicology, all your in vitro studies to make sure, and your laboratory animal studies to make sure the drug's safe. Then it goes into clinical trials when you, in other words, when you're working with you're putting the drug into human beings. And eventually you get permission to get your drug onto the market when you've demonstrated that you could provide it in good quality and it has good safety and efficacy. So that's the schematic for the pharmaceutical industry, but any other industry has got similar stages. You've got the research, discovery, then you might take out a patent, and then you do development of the product and do full testing to make sure it's fit for purpose. Then you get permission to put it on the market. And the kind of theme behind, so this is, I've seen 9 or 10 at least different schematics of getting a drug to the market. This is one which I think came from AstraZeneca, and if I was going to do it myself from fresh, probably for me the major decision points here when you decide what of these active compounds, which one to put in preclinical development. This gap here is where you start going to humans, and everything from here is regulated. So I would say that's, for me, that's the, well, when I was in industry, I worked in this area anyway, and picking your active compound to put it in development was always one of the key decision points. Right, so that's one of the ways that the pharmaceutical industry is depicted. I have my favorite way I like to sort of show it because it emphasizes the role of analysis, and this is what I'm trying to get over today. All industry is market-led, and it's cyclical. It's not just you start in discovery, you go to the market. It's a continuing process. So if it's a market-led company, you get the ideas, where are the unmet needs, where are the large patient numbers. Then you decide what therapeutic area you're going to work in. Then you have to look into the biology of the disease and see how you could intervene in the disease pathway. And once you've decided that, you've got a target, you might want to make an inhibitor, something will block the target. Then you have a lead compound from range that you might get from studying the biology of the disease, and you want to make lots of compounds till you find active ones, and you try and optimize that before you go into preclinical development and get the best drug-like properties, not just the most active compound. But a key point here is once you've got some active compounds, you make a few analogs to make sure that you've covered all the kind of structural variations that you need so that you could take out a patent, like Dr. Sacha's patents, and that means that your competitors can't make the same kind of compound and copy your invention. So when you take out that patent, in the UK, you have 20 years exclusive rights to your invention, which means if you've got an active compound, you need to get on the market quickly because you only got 20 years. So from this point here, from going from kind of discovery to get through preclinical to clinical development and getting regulatory permission to launch your drugs, this could be 8 to 12 years. So if you've got a patent the last 20 years, you've only got about 8 years or so, 8 to 10 years, 8 to 12 years left to have exclusive rights to your invention and sell it without any competition at a price, not to make a unfair and profligate profit, but you need a lot of money because this research is very, very expensive. So you need to make a lot of money here so that you could build up the funds, not just to satisfy greedy shareholders or non-greedy shareholders, but to plow back into research. Because, as I said, this is a very, very expensive business, and you need to have companies prepared to take that risk and get suitable reward. Otherwise, we simply won't get any new drugs. So there are all these different stages, and what I hope to show is that in an analysis, there's a strong interaction with each of these stages, which is important and could enhance the knowledge and lead to useful information about your drug and your drug product. But because you have all these interactions, the interactions have to be very quick, and if the analysis is slow, it slows all this wheel around, and the slower the wheel turns around, the kind of less time you'll have left here to benefit from your invention. So I'm going to reiterate, I'm going to try and show that analysis has a positive influence on all these different stages, and that we could speed up the analysis to make it much more effective, and therefore help the whole operation of getting drugs to the market more quickly. Right, so now the first example, as I was about to say 2 slides ago, is actually an example not from the pharmaceutical industry, just to show that it's very widespread. Even when you get into manufacturing, the analysis could help get you better yields and a smoother process. Now this goes back in time. Polyurethanes were, and perhaps lost interest in how sofas are made, but a lot of furniture contained polyurethane foam. It's a major commercial industrial product, and to make that you take an amine and you will use phosgene, COCl2, to make an isocyanate. Then the isocyanates get polymerized to form the polyurethanes. So the problem is that if you're doing this industrial scale, this is very, very poisonous. So it's a danger to people around your plant and also to the people operating the plant. And it's one of the ways isocyanates used to be made in industry. So this was a project where we were looking at an alternative process where you took a nitro compound and reacted it with carbon monoxide, which, as I'm sure you'll be aware, is also highly poisonous. So this is something I did way back before health and safety were such major issues, and I routinely was working with 180 atmospheres of carbon monoxide. It was in an autoclave, but I had to vent the autoclave. So I was also working with metal carbonyls, which are very good. So I'm lucky to be here today because I was working with some fairly dangerous compounds back in the day. But anyway, this is an awful lot safer than this. And this process had a lot of potential to replace this traditional reaction of phosgene with primary amines. So there are patents there showing that was a high yielding process, showing about 60–70% yield of isocyanates. This was phenyl isocyanates. The actual ones used to make the polyurethane is MDI, methylene diisocyanate, and toluene diisocyanate. So I did some of this work as a secondment in industry, and I had to learn a different language. They're talking about MDI, TDI, so I had to remember all of these acronyms. Anyway, so the idea was to try and optimize this kind of process, improve it so it was higher yielding and had commercial potential compared to the traditional one. So when we did that, we were looking for 60–70% yield and then trying to improve upon that. What we found was that it was isolating isocyanates, and from multiple reactions, very disillusioning. For a young student, I was getting a black tar every time. And when I went to use gas chromatography, I found that the yield wasn't 60, 70%. It would often be kind of maybe 7% or 20% on a good day. And if I was really lucky, about 30, 40%. So either I wasn't doing it right or there's something wrong with the reaction. Now, this is where HPLC came in. It's when I first started doing HPLC, and it made a major difference because a lot of these products here, like the triphenyl biuret and the urea, you've actually got protons there, and they come from the reaction of isocyanates with water. So a lot of time was spent trying to eliminate water. So on GC, we're getting a variable yield, and also isolating the isocyanate yield was probably worse. So when we used HPLC, the isocyanate would react with silicon and HPLC column, so we'd kill the reaction by reacting this ethanol to get the carbamate. So we're analyzing the phenyl ethyl carbamate by HPLC. And when we did that, we not only were able to quantify that without putting it into a GC at very high temperature, we were able to pick out all the reaction byproducts, and then by having a device, a fine capillary to put in autoclave, we're able to follow the reaction with time and see how, not just follow the product, build up the product with time, but see what was happening with all the byproducts. And what we found was early on we were getting the urea and then the biuret here, and with time these actually started going down, and these cyclized reaction byproducts were forming. So all of our work had been around about trying to eliminate water from the reaction, but what we saw was these products were forming, and what we did was isolated them, and we suddenly realized we kind of put them down the GC column at a high injection block. And what we found was all of these degraded under heat. They're subject to thermal degradation to yield the isocyanate. So what we're seeing was a little bit in our GCs, a little bit of free isocyanate, but most of that isocyanate has been measured by the GC, by the competitor, was actually coming from the byproducts and not from free isocyanate. And that explained when we were doing isolation, we were doing room temperature vacuum distillation or at high temperature, and the GC, we'd have a heated injection block, different column temperatures, and that all led to variation in isocyanate. So when we tried to block water out completely, the reaction simply didn't go. So we discovered a lot more about the reaction. So the idea is to add kind of traces of water to actually generate these rather than keep water out. But whatever way you do it, you're still getting a lot of these. So in a nutshell, we found out why it wasn't such a good process. And if we were going to take this further and optimize the process, we'd have to get the right amount of water and the right amount of workup to get the maximum yield of these isocyanates. Okay, but this is a major problem for this UK company trying to compete with their American competitors, and we're able to work out why the reaction didn't work and how we could get a consistent high yield. Okay, and it kind of solved a major problem for that company by using GC initially, but the kind of killer was HPLC. We totally worked out what was going on. Right, this takes me through to a more recent history, but still kind of in the past, but nevertheless a very important one. And this is development. This is the last one's contributing to chromatography, making a critical contribution to solving a manufacturing problem. What I want to talk about now is another real example, not an artificial academic example, of how chromatography solved a really major problem in the drug industry. So this is a compound called chromokalim. which is a cardiovascular drug, can be used to reduce blood pressure. There's quite a lot of excitement by this compound when it was going through the development because it's a new mechanism to treat cardiovascular disease. So there's a lot of interest in it, and the company was very keen to get this to the market quickly. And the original compound going into development was this molecule here, this amino alcohol. And what one of the discovery chemists actually noticed was that when this was administered to rats, there was an incubation period. It was only after 30 to 40 minutes that the blood pressure kind of went down. So he thought, well, maybe this sort of time lag might mean that the drug, the candidate itself isn't so active, but it's actually a metabolite that's active. So they collected some rat urine and extracted out all the exogenous compounds, but also all the drug and drug metabolites that are in there. So we analyzed it. We found very little of the residual compound which had been administered, but we did find all of these metabolites. So by using semi-preparative HPLC on this rat urine extract, we isolated this compound, and from this these other metabolites. Okay, so, and we isolated enough to be tested in rats, and sure enough, this was instantly active and was obviously the active species of the compound in vivo. So what happened was we changed the course of the development of this whole project, and this was replaced in development by its active metabolite, which is chromocalim, which got to the market in Japan, and it's not still in the market for reasons I won't go into. The problem was that it's wasn't sufficiently selective, but these potassium channel inhibitors are still used. They found an interesting side effect that actually, I think it's minoxidil is a potassium channel inhibitor blocker, and it's used as a hair restorer. I believe it's called Regain, contains minoxidil, I think. Okay, so, but it was an exciting compound, and we did help it get rapidly to the market. We isolated all these metabolites, but this was the main metabolite, and it was very active. So chromatography almost discovered this exciting development compound, which was highly rated at the time. And not only that, you notice that this drug has got trans stereochemistry. There are 4 stereoisomers about here. The active species, the trans isomer, cis is an impurity, but you've got 2 enantiomers for the trans. And if we look at this particular separation here, so it's a racemate, and at the time, a lot of synthetic drugs going into the market were racemates, but after the thalidomide tragedy, all the regulators were acutely aware of the potential problems with racemates. But it was only till the late 1980s that the technology became available for actually studying the racemates together and separately, and all the regulations changed, and that drugs had to be, the regulations changed about 1994. The FDA put out a position paper, 1993, a position paper in a journal called Chirality, essentially saying that you should look at both enantiomers individually and pick which one was the best, and if you're going to use a racemate, you had to justify it. So at the time, we could see that this was coming, but the regulators couldn't do anything about it. It was only with the development of chiral HPLC stationary phases, was it possible to separate enantiomers? Now, they've got identical physical-chemical properties in an achiral environment, like in a C18 HPLC column, and it is a difficult separation, but once these chiral stationary phases improved, the big development was when a plasma protein found in the body, alpha-1 acid glycoprotein, was immobilized onto silica. Now, it's much better than previous HPLC chiral stationary phases in terms of the range of compounds it could separate. We're able to take a lot of the development compounds on our site at the time, and for what had been difficult separations, we could get really, apologies of struggling to find where my cursor is here. That is an enormous selectivity in terms of alpha that we use to measure separations. Often you're talking about between 1.1 to 1.4 if you're lucky. This is an alpha value of almost 4 or 5. That's a phenomenal difference in selectivity. This is only an alpha of 2, but still quite good. These are all the lead compounds coming through the various projects at the time. So for the first time, these could be studied, the single enantiomers could be studied, and that was a major breakthrough. And this is a separation we got from chromocalin. It's not pretty, but allowed us to further look at studies. I think, have I got the next? Yeah. Sorry, I thought it was someone from India calling to say we can't hear you anymore. It's my dentist phoning about an appointment. So I'll be going to the dentist on Friday. Right, where were we? Yeah, so this was a good enough separation for us to be able to look at, again, going back to rat urine, and we were able to kind of study what happened to the different enantiomers over time in the body. So we looked at rat urine, dog urine, and also we pooled enough human urine to look at human urine as well. And this hadn't been possible before. And we were able to find out that the second peak here, this one of the enantiomers was metabolized very quickly, and the one which persisted and was responsible for the activity was this first one here, which is levochromocalin. So had things gone better in Japan, chromocalin would have been followed onto the market by levochromocalin. So we've totally transformed the progress of drug candidates going through to, not just for chromocalin, we also did chiral separations on paroxetine as well, and just about all the development compounds are racemates, and we're able to get kind of separations such as these. Paroxetine was a different case because it's always known that the receptor site was very stereoselective, so even in the beginning, paroxetine was one of these compounds which had always been made as a single enantiomer through chiral chemistry rather than making the kind of racemate and then picking which was the best one. Okay, so that was a major impact in development in the pharmaceutical industry. So at the time, we're also working in discovery and sort of collaborating with chemists. So I could do a whole 3 or 4 lectures on all the different applications we've got. It's a very exciting time working with pharmacologists and synthetic chemists in discovery and helping them study how their drugs were working, doing some difficult separations, axial equatorial isomers, doing this in the PrEP scale, and beta cell tropin. That was about there's a peptide. We did some semi-preparative HPLC, and that was used as a surrogate model in obesity projects and diabetes projects. And we can develop some assays so that the synthetic chemists and biochemists could monitor these markers for how well these drugs were working. But also, we're able to interact and even help with some of the synthetic chemistry. I'm not going to talk about this one an awful lot, but you might recognize this compound here. It's a beta-diketone, and these kind of reagents are used with europium and lanthanides to have lanthanide shift reagents in they're used in NMR to give you, and you have chiral lanthanide shift reagent. So the idea was to use that in chromatography. Now we're looking at the wrong mode of chromatography, so it wasn't progressed there, but I'm still going, even many years later, I'm still going to revisit this and use it in a different mode of liquid chromatography, so it still has some potential. But what's important is that we're working with synthetic organic chemists making drug molecules. So we had this immobilized. So we found, and there's a ChemCom paper on this, that we're actually contributing to the progress of synthetic techniques, because this was at a time when it was extremely difficult. This was a very, very good asymmetric synthesis catalyst for the addition of carbenes. Okay, so we're supporting the discovery chemists, and also we're helping them produce better ways of doing the reactions, all through developments in chromatography. Right, okay, so I'm going to go back to manufacture for getting purely on chromatography and speed in chromatography. But I'll say a little bit more about speed to the market before we do that. It's going back to this wheel, just to reiterate. So the original example from manufacture was not from the pharmaceutical industry, but I'll show you how the speed of analysis is important in terms of manufacture and supporting manufacture. Okay, so this is a real example again. This is supporting MSD in India. You probably know that as Merck. In the UK, they're known as Merck Sharp and Dome. And this is a part-time PhD done by a student who's working for an employee of Merck Sharp and Dome. And they have to do something called cleaning validation. You don't have one piece of equipment dedicated to one industrial process. You might make a big batch, then you'll change over to a different compound. So when you're doing that, you don't want to have the previous compound being manufactured on the equipment contaminating the next drug you're making. So you have to do cleaning validation to validate that your cleaning processes are cleaning up the equipment properly. So on a major kind of manufacturing site, you might be working with 8, 9, 10 different drugs. And if they're not very water soluble, it's very difficult to get them off the equipment, and you have to pick up trace levels of residues on the equipment that's poorly soluble. So it's quite challenging analysis. And if you're developing a different method for each single one, it takes a long time. So what you can do to be more efficient, speed things up, is have one generic method. So this is an example of this part-time PhD student did, and we're speeding up not by the speed in the chromatography, but the speed in the method development. This is the kind of thing that's fairly routine now. We had, I think it's about, I've counted the peaks, there are about 10 peaks there, which were the worst case scenarios, the ones which were most difficult to clean. And we got this one gradient HPLC method. This